Well, welcome to the 19th session which is about conceiving about publishing regulatory matters in nanomedicine. I will be the first speaker, and welcome to this session. That these are things that we never get really into complete understanding, but they have to do again and again, like the regulatory bottlenecks, what is the ethics relations, are we doing a real thing or just something that looks real to us because we are always in the middle of it. And of course, they are very much interested in how this whole thing could be managed. So let's go and start with the first thing. Like, I'm going to talk briefly what's behind the controversial directions of science and whether are they controversial and what we're going to achieve in precision nanomedicine. So what is science? Let's start there. The beauty is that it means different things for different people. That first of all, it's a systematic study. Then it's an organized body of knowledge. Also we call science, and it's a systematic enterprise that builds and organizes knowledge. Recent publication titles give us a warning sign that those one that how science goes wrong, science of fake news, spread of true and false news online, identify pseudo-journals, and even a shocking title that why most published research findings are false. So something is not really right. And we all experienced the flood of unverified information on the internet, the complete lack of self-policing and quality control. Then also sensationalism in newspapers, magazines, and sometimes journals as well. One of my colleagues called it's like a fashion show when you see beautiful clothes that you would never wear on the street. Of course, without basic science, there is no applied science and so on. But the worrisome is that there are studies that cannot be replicated, and not even one of them. The Royal Netherlands Academy of Arts and Sciences come up really 2 years ago with the request that make replication studies a normal and essential part of science. So before I go further on, let me explain to you my view. How do I see the hierarchy of what we know? So why don't we know and understand everything? We are all surrounded with hearsay and accidental observations. You go to work, maybe not right now, but in general, go to work in the morning, you meet your friend, you see an accident, you remember those, and you don't remember the other 1,000 people you've seen in cars, and because our brain filters that information. To really understand and really have knowledge, we need data that are observed and measured, and we need to understand. And the more we understand that, the more we connect them, they are much more useful for us, understanding what's really. So from the data, when we understand the relations between data, that leads us to an information. And information is to change somebody else's opinion. And we have to understand the patterns between those pieces of information, which takes us to knowledge. This is kind of passive knowledge because we need to understand the principles to get to the stage which I call wisdom. And the wisdom enables us to take intelligent action and simplify complexity. And the most vivid example is Einstein equation. Could it be more simple? I don't think so. But how much time it took, and how hard was the road to that simplicity. And my point is that the essence is always simple, only the details are complicated. Every component in the scientists and the interests of scientists and organizations and the publishers are deeply interconnected. And in an ideal world, everybody is happy. Of course, there is no such a thing, an ideal world. And recently meaning that the last 25 years, business is blooming, but people are not really happy. And the science is some of them yes and some of them no. But what we really have to take into account that these interests not only coexist, but cannot exist without each other. Therefore, everybody has to adjust a little bit to avoid the variation of the whole. Negative trends appear, throwing off the balance in our world of increasingly virtual reality. We have this virtual conference now, but we run Zoom meetings all over. Because of the pandemic, it's even more complicated. And these are just a few of them that authors are forced to increase constantly the number of publications. And some people do more works, others become copying, multiple submissions, minimal publishable information, and so on. Meanwhile, the publishing businesses, since the open access models introduced, they shifted their financial risk to the authors. So then they get paid, and that's pretty much it. The opportunity to make easy money have led to predatory journals, bogus conferences, and I keep saying and I keep advertising that Clinum is one of the best networking meetings, because it is. Differences between basic research, academic research, R&D, clinical medicine are being blurred more and more, including commercialization. And it's true that research topics are way more complex than they were 10 years ago. And it's also true that for research groups, more money is available, but less number of research groups. Network computers have generated entirely new opportunities. Online journals require only hardware and software, distributing information online much more efficient, archiving easy, retrieving, storing articles. Everybody has PDFs with the thousands in their computer, and which actually is searchable. It's very, very nice. And customized information is also available, which just makes it convenient for the first look. Now, by 2012, 2014, the capacity to store information in a digital format, it was, and we are at reading 2020 now. So everything is in digital. Now, digital storage of data, unfortunately, now allows the manipulation of data, which you couldn't do before earlier. So easily, and the injection of alternative facts, especially in the US, create distort false information, which may result in a personalized world for us to live in. AI algorithms collect your data to learn your preferences and give you a personal feedback to confirm and reinforce your beliefs. Let me give you an example. Go to video lunch, not now, after my talk, please, and science. And the first one is, it's a 3.5 minute video about precision nanomedicine, Pierre Nano, the official journal. Now you watch it, and this is what happens. I did watch it. And I wonder what will you see, or who will you see, because this is what comes up afterwards. That's John Oliver is promoting precision nanomedicine. What happened, that the algorithm of YouTube changes the picture to one of your favorites, and even better, because the next one when I watched it was like the 25 best jokes of the weekend. So it doesn't really matter. My point is that different people may not see the same data or may not have observed the same facts, quote unquote, which generates conflicts. And human nature is have two ways of resolving conflict. Actually, three ways. One, you avoid it completely. You don't talk about it. But when you talk, there are two ways. Or what are you saying is wrong, or you are stupid. And these two things generate very non-scientific discussions, or may generate as well. So there are subjective and objective reasons that create these problems. First of all, I look at it from the part of different interests. And there are certainly constant conflicts between interests, which have to be resolved from time to time. That is, the individual is part of every individual part of a family, then it's a second group and part of the third group, and that builds a society, and countries' interests are different. The global interest is different, again, and definitely may be interest from the individual ones. And there's a vertical I call haves versus have-nots, funded research versus unfunded research. A student or a postdoc has a different interest than a tenure-track faculty, and the tenured faculty have again different interests, and so on. Second issue is like, as I see it, having publications became an existential issue in academy. Universities, I've been to a few of them, they don't take the best guys, they have those, prefer those who have the most research money available. So the 5-year grant, in the middle of the 5-year grant, you can transfer $1 million, you have a very good chance to move. Penetration of politics into science, and definitely in the US. And why it is problematic, because science would be based on facts and logic, but politics is based on perception and emotions. It went so far that recently the National Academy of Sciences president and the National Academy of Medicine in the US, they issued a statement about this political interference amid the pandemic, that they find ongoing reports and incidents of politicization of science, overriding of evidence and advice of public health officials, and any effort to discredit the best science threaten the welfare of us all. A subjective thing also, that's true, but political systems fund science through different mechanisms, centrally, through proposals, or grants, or both. Grantees better satisfy funders. No optimal method, but it's some kind of combination. And there are some objective reasons for lame papers. Graduation or receiving a PhD does not make you a successful author. People must learn the art of communication, and it's part of, and at the basic level, it's everybody and anybody can learn it. But it's certainly an art. Not everybody is a great writer or a great poet. And the second thing is, which is objective, cutting edge science is always fuzzy. We all the time argue what to measure, how to measure, why, relations, patterns, principles, actionable science. Remember, data, information, knowledge, wisdom. And they need to understand the relations, pattern, principles. Actionable science comes much later. So there are a few issues we try to address. The replication studies, we accept replication studies, that's verifying research data. Negative results, because it's very important to need to not only what to do, but what not to do. Eliminate the limitation of paying excessive APCs. Not everybody has $2,000 US to pay for an article. And reduce long waiting times. So the way we envision this, that's our requirement to share reliable information. Plan and data must be good, discussion logical, and all the conclusions are open to interpretation. And members of the editorial board and other reviewers, we invite expert reviewers, they help interpreting that. So we assist authors instead of criticizing them. This is the homepage of Precision Nanomedicine, which is a peer-reviewed international journal. We launched it in 2018 April, end of April, and we have our strengths are the 4 Ps: people, publisher, policies, and procedures. For people, we enjoy the support of world-renowned scientists, including the CLIDAM community, the European Society, and the International Society for Nanomedicine. We own our publishers, so we're not a publisher dictator. We can set our own policies the way we do the best. And we help authors to publish good articles, not reject them. Procedures, we have fast processing times, and almost everybody works for free. I have 14 plus years of experience as editor-in-chief, and Georgette, our managing editor, has 7 plus years of experience. We have 2 clinical editors, a practicing clinician scientists. They write the significant statements that something is basic research, potentially clinical significant or clinically significant. The team of associate editors has been working together for years now, and everybody has a specialty, but also have overlapping experience. Members of the Honorary Board are our strategic advisors. They have a different view. They are very busy people in the different view of the field. And that they make sure that the general direction of the journal is correct. We are truly international. Strength number 2, that we own a non-profit publisher, and this is the publisher's website, endoverhouse.org. There are no submission fees or APCs as long as we can make it. Authors retain the ownership, including commercial use, and published articles are free to download for everybody. This particular example had received more than 5,000 views and 2,000 downloads. The strength number 4 is procedures. Publishing an accepted article takes only 1 week, assuming that someone sends the PDF corrected galley back in time. Abstracts of published articles are posted to 70 Nanomed-related LinkedIn groups, Twitter, and so on. We have accessing about 500,000 people weekly. Of course, not everybody is interested, but the field is so broad that it's impossible to target. However, we email to 1,300 members of our core group. Anybody can be a part of it. Just drop me an email, and these are coming out weekly. This is an example. So PNNO keeps growing. We have published up to date since in the past 2.5 years, 50 articles, 57 citations, a lot of visitors. The growth is illustrated since October, illustrated the best by looking at resolution attempts, which are clicks on a DOI in our articles that we publish. And in summary, we are a journal you can trust, no publication fees, fast processing times, and you can submit your work anytime at precisionnanomedicine.com. And if it's too complicated, you prefer a shorter URL for the first time, just come to prnano.com or write me an email, volunteer to our Precision Nanomedicine PNNO group on LinkedIn, and thank you for your attention. I'm back. Thank you, Eric. Now that's the next one. See, I made a mistake. Rosie is the next one, the Nature Biomedical Engineering, and will talk to us about a very interesting topic based on the research. Rosie, please go ahead.
5, 4, 3, 2, 1. I'm not sure if I should be counting. I did anyway. So thank you, everybody, for being here. Thanks to the organizers for inviting me. Today I'm going to be talking a little bit about what we do at Nature Biomedical Engineering, where I have been an editor since before its launch, actually. So we launched in 2017 and we started the journal in 2016. So today I've been asked to talk a little bit about how can we add feedback to our authors, to our reviewers, and to our publishing process, and how can we insert some of the regulatory bottlenecks that nanomedicines face during primary research planning. So hopefully you can see my screen move, yes. So NBME focuses on technology for human health, and I think that is a very specific fingerprint that we give to the articles that we publish. We consider articles from the engineering, chemical sciences, material sciences, chemistry, physics, but they are all there has to be an element for improving or understanding human health. So we look for a degree of advance, which is conceptual, fundamental, methodological, technological, and we focus a lot on the translational value of the work that we publish. So this means understanding human disease and improving healthcare. Of course, the articles that we publish need to have breadth and depth of study, and I will discuss a little bit about that in my next slides. So because we focus a lot for technology for human health, and I said we are a little bit different from other journals, certainly within the Nature research family, because we are an applied journal that looks at boundaries between fields. So we look at discoveries that are at the interface between medicine and the physical sciences. So what do we look for in articles that we consider for review? So we look for the breadth of the study. So we need to be convinced that the conclusions would be of interest or relevance to scientists within a broader field than those specifically very close to the field of research of the article. There has to be a conceptual advance, although not only a conceptual advance, because obviously we look a lot at the translational value of the study. So it is either a conceptual advance or a performance advantage. Of course, there's no secret formula that would guarantee either publication in our journal or even sending the article out for review. In fact, our decisions are based on evaluating each manuscript that we get and looking at findings and whether or not the data support the claims. So how do we really look at translational relevance? You know, what does that mean? And I think I was trying to think of ways of discussing this for this meeting, and hopefully I will be able to convince you that what we're doing goes in the right direction. And I want to discuss a little bit about experimental design, because I think that really is the key factor for good work. So great papers need to be structured around a question that takes into account factors that have implementation in mind. And this is because we know that the barriers to clinical adoption and implementation of innovation in healthcare have a practical, methodological, and also an economical side to things. So we would like to see certain elements of the implementation aspect of the work addressed as much as possible early on, so that when we get manuscripts, those elements are there to be evaluated. They don't necessarily mean that research is less interesting if there are a series of limitations, but we do ask for transparency of those limitations. So also, of course, we understand that funders and other business stakeholders in healthcare, so whether it's big pharma companies or venture capitalists, need to recognize that implementation efforts have to start earlier on in the research process. So there has to be elements that in the early stages of research that are going to facilitate the implementation of the research. So we recognize that innovating clinical practice can't be an afterthought. It has to be the main goal. So I put here a couple of articles that interpret what for us, what we can publish that delivers that translational aspect, and how do we go from animal models or from cells into to larger animal models, non-human primates, and in humans. So this is just 2 examples of articles where the experimental design was delivered through multiple models, multiple animal models, and indeed multiple species. So now I want to just briefly talk to you about the double diamond structure for translational research. That is something that I find that I use a lot. It helps me understand how to place within the same framework preclinical, translational, and clinical research. Now, the double diamond, I don't know if you're familiar with it, but this is something that comes from design and engineering sciences, but is, I think, not very often used in academia, certainly not the venues that I have been to. But I will just briefly talk you through it. The double diamond is a design model, and it's a tool that you can use for a systems design, essentially. And so you can use it to understand how different stages of research can fit under the same framework. So the first stages of each of the diamond, which are the discover and the develop stage, are divergent thought and hypothesis testing. So these are typically experiments and research that is dedicated to either gather knowledge or test a variety of potential solutions. And then you have the second stages in each diamond, which are convergent phases of thought, where you can really focus and test workable solutions. So the way the double diamond works is that you start with identifying a medical challenge. You have a problem that is close to your heart that you want to solve, and you iteratively gather knowledge and define the focus area that you want to define a problem on. And then once that problem is very clearly defined, you can then try and frame the solution by developing, again, iteratively, a series of solutions, and finally deliver workable and testable options to finally get to something that hopefully is a solution that is a deliverable clinically and clinically valid output. So once you use the double diamond structure, you can get sort of two overarching themes. One is for disease prevention and the other is for disease management. So disease prevention means that all of the stages of research, that divergent and convergent thought, can be oriented to expanding our understanding of a disease and actively acting preemptively to stop something from happening. And so this is knowledge on the mechanisms of the disease, but can also be, for example, early predictive biomarkers for a disease, where you would do a test very early on for, let's say, asymptomatic people, and avoid progression to later stages of a disease in cases of where disease is diagnosed late. And then you have obviously disease management, which encompasses, broadly speaking, your diagnostic tools and your therapeutic approaches, basically trying to stop a disease. So both prevention and management themes can include biomedical and clinical research, and they provide entry points for the regulatory bodies, because the regulatory bodies hopefully can look at the different stages along the pipeline and can identify what problems there may be later on. So because the double diamond is an iterative process, the whole trick is to look at the later stages and try and identify problems that would come at a later stage and introduce them into your, you factor them in at an earlier stage of research. And obviously doing this facilitates discussions of general interests, such as vaccine safety or efficacy of different therapeutic interventions. So how do we actively use this kind of framework in our publishing? We look for whether or not the intended use of a nanomedicine device has been identified. So I think this is a really key factor in the experimental design. There has to be sufficient evidence that the proposed research is going to be necessary to solve a problem. And what is that problem? And is the experimental model that has been chosen appropriate? So do you have, are there enough animal models? Does the work require multiple species? And has it been tested in human tissue? Or in the case of devices, it's easier to do. You can test the device in healthy subjects or patients. Do we have unprecedented design and functionalities? And this is the point that I think we look for, certainly in manuscripts. Having said that, I thought I would put it black and white, that formulations that we see are overly complex and procedures that are very complicated, that require too laborious, we wouldn't necessarily class them as better. So we do look, I think multiple people in the previous talks have listed sort of simplicity as being, you know, a positive factor rather than a negative one. We look for head-to-head comparison of our technologies that we evaluate against state-of-the-art and clinical standards. So when we're looking at performance, we really want to see performance against the best in class. So whether that's the therapeutic standard of care. So we really want to see the design of suitable controls, not just whether or not the delivery of, for example, a drug in a nano formulation is better than the native drug, but does it perform better than the best in class? And then we look at the protocols and methodologies in the paper, and obviously look at whether or not they are suitable. So the suitable molecular biology techniques, is the sufficient mechanistic data, or the validation experiments good enough? So we put these together, and I list here sort of like the SMART criteria. I guess some of you will be familiar with them, if not most of you, in terms of what makes for specific, measurable, acceptable, realistic, and timely criteria. So we try and evaluate these, for example, and introduce these elements in our evaluation criteria. So if we're looking at reduced systemic toxicity, for example, we look at how is this shown, which organs have been evaluated, what is the time frame for the toxicity studies. When we see, for example, a manuscript that says, "Oh, this formulation does not have any toxicity,"
you have 2 more minutes.
Thank you. Do we see this in terms of weeks, or has there been multiple months where this has been evaluated? Is there complete biodistribution data? Do we have sufficient dose response data, route of administration? Has immunogenicity been tested in an appropriate model? And when we look at improved targeting, we're looking at specificity data and how good is that specificity data that's been given. Have the authors shown off-target effects? If there is, and in cases where there is a clear increase of the payload at the target, how better is it, and what defines that better? And all these elements necessary in the clinical for the work to be translationally relevant, or are they simply nice to have? And then another important point is whether or not there's been any data omitted from the study. So we take this quite seriously, and we suggest that all our articles, all our authors deposit the data in public repositories. So a little bit of examples of published content. We recently published a comment article on covering the EU regulation of medical devices, and these specifically look at new regulations that are coming into play this year for the MDRs and in 2022 for the IVDRs. Again, we look at a focus on implementation in the work. So this is another article that we published that looked at the opportunities and challenges for implementing optical imaging biomarkers, for example, and the number of feedback elements that need to occur between different points. And with this work, we really want to highlight the importance of technical and biological validations and clinical utility assessments, so that these implementation bottlenecks can be identified and solved early on. How to surface content from NBME? So if you go to our website, you'll be able to access our collections. And although there's dates on them, these are updated on a regular basis. So for example, you'll find the delivery of drugs and biologics has a lot of nanomedicine work in them. And finally, I just wanted to finish with some examples of published material where we looked at, for example, biomanufacturing, cell therapies, how to design preclinical studies in nanomedicines, and some examples of articles that we published. So that has basically me done for this talk, but I just wanted to sort of summarize that how do we incorporate this knowledge on the regulatory bottlenecks in primary research? We ask our authors and our reviewers, often before review, to address these potential limitations in the study. We use the best information available across translational space to really set selectivity criteria. And we try and focus as much as possible on selectivity criteria that look at how the experimental study design was done. And we look at work that has implementation in mind. So thank you very much.
Well, thank you. Thank you, Rosie. And let's listen to our next speaker, who is coming to these meetings. Like, since I didn't come for the first one, but Don was always here, and he has a very interesting background. He has a degree in chemistry and theology, and I worked extensively from related technologies and nanomedicine since like 17 years now. Don, go ahead. You are our ethics person.
Right.
What about the mini robots, the nanobots, if you like?
Can you see this?
They sound a bit like a robot, a bit like a drug.
Are we up on the screen?
Yes, I.
Slide. Okay, can you see this?
Yes, we see. I
Right,
see.
okay. Well, thank you very much indeed, Lou, and thank you especially to Beat for this whole conference. As Lou said, I've been taking part in this since almost the first ClinAm, but I explain my rather strange background. I trained as a chemist, worked in the nuclear energy field for many years, then switched to ethics and worked for the Church of Scotland for 15 years, running a unique project on ethics and technology. But since 2007, I have been running my own company as a consultant called Ethics Limited, and I've been involved in something like 6 European Commission research projects on nanotechnology and biotechnology, usually in the role of an ethicist or in the role of dissemination with publics. I should also mention that I am currently a member of the United Kingdom government's Animals and Science Committee, which involves a lot of very interesting other ethical issues, some of which relate quite a lot to what we're talking about in this conference. Well, we've been talking about bottlenecks, and there is another bottleneck apart from the regulator, which is society. Here is the Queensferry crossing, the new bridge across the River Forth, just north of Edinburgh, and there's a barrier between technology and society. Novel technologies in the biosciences and nanosciences, the research happens far from the public view. Although patient groups are increasingly involved in medical translation, if you're not actually a member of a particular patient's group, the chances are you know nothing about what's going on. Innovators assume the public will want what they produce, and if it's not familiar, well, we just need to educate people. And for conventional medicine, that's a pretty good assumption. If we're ill, we want something to make us better. But novel innovations, particularly when we start really digging down into the fundamentals of the body, of cells, embryos, genes, and anything to do with the brain and the whole brain-machine interface, we can start getting some, it starts to get a bit scary ethically with the public. And so what if publics don't accept what we're doing? My background in nuclear power, I wrote my first article about the ethics of it in 1976. But that also irradiated food and then genetically modified food all challenged the assumption that the publics would accept any technology which science and industry and governments invest in. And in 1999, the GM food crisis, in which we were heavily involved, because we have been working on this field for many years before, caused a major rethink about public acceptance. And there was a shift from we must educate the public to engaging publics in a dialogue. And the concept of upstream engagement became the buzzword of that particular period in the early 2000s. We consult with the public much earlier about the direction of a technology, even perhaps the research itself. And nanotechnologies were the next new technology pipeline and became the subject of unprecedented public consultation. Science policymakers feared that nanotech might be rejected as another GM. This was a photograph taken by Patrick Boisseau from CEA Grenoble, and it was at the occasion of the inaugural meeting of the European Network of Excellence on Nanotechnology and Biology called Nano to Life. And somewhere on the wall outside Barcelona University was this graffiti, which I understand to mean Nano to Life, technological fascism. So rather than being the next industrial revolution, people are actually afraid of what the revolution might cause. we did a study in that Nano to Life project, which was the very first time we had really looked at the ethics of nanobiotechnology in general and nanomedicine in particular. And one of the things we found out was it wasn't so much that it was nano as what it was enabling that raised the ethical issues. So in a way, it wasn't so much the nano part of it as what it enabled medicine to do in future. So for example, in diagnostics, we had the lab on a chip idea that you would just go to your doctor and say, well, I've got a cough, and he will be able to take a quick blood sample and find out very quickly a genetic analysis of you. So you would know which particular antibiotic might suit you. But of course, he might also be able to find out that you've actually got all kinds of unidentified genetic problems, and suddenly you've got real problems facing you later in life. Do I want to know that? So the question we then asked was, actually, are we all ill? If you could analyze all the biomarkers that nanotechnology promised it could find, we'd all be ill, wouldn't we? So what now was being well? So there might be questions about what we want to know about in the area of diagnostics. So it wasn't so much the fact it was nano, but what nano enabled. Well, we started to engage with the publics, and the idea of manipulating these forces and entities at a small scale was actually a bit of a disjunction. For the scientist, great new powers at her disposal. She could do all kinds of things she'd never done before. Unsettling for wider publics because it really is safe to be doing this fundamental stuff that we've kind of only vaguely heard about. And it turned out the engagement wasn't very effective. Nanotechnology was much too diverse a subject. There weren't enough examples, and the outcomes were really that were a new awareness of possibly risks from nanoparticles, a concern not to be too smart with your devices in the body, and being wary of commercial drivers. But there was a lot of support for nanomedicine and a certain amount of doubt about nanotechnology in food. The EC developed a concept of responsible research and innovation coming out of all this. The idea of how do we govern the challenges of emerging technologies after GM food. The idea was that rather than just have the usual suspects, science, industry, and government, we should also involve the public as more than just consumers. They should engage with the proposed innovations as citizens in some sort of collaborative way, because publics might hold values, concerns, and insights that are different from those of the technologists, and may indeed help them to design things that they really want rather than didn't want. So responsible research and innovation should have the power to challenge what the usual players would do if left to their own devices, as it were. It should anticipate the impacts, both intended and unintended. It should reflect, cause people to reflect upon what are we assuming, what are the purposes, what are the uncertainties, and then to engage with publics to widen the vision and be prepared to act in response to what the insights from society happen to be. And that can be quite a big ask. The NanoAthero project, which is on the logo on the top of this screen, was a project which I was involved with between 2013 and 2018, looking for nanoparticle systems which would be able to identify unstable plaque in atherosclerosis and or blood clots, with a hope of helping to address what is actually the major cause of death and disablement in Europe. This came directly out of Clinander's project, and there was a decision that they wanted to look at the ethics, so they asked me if I would do this. And there were a number of issues which we were obviously looking at from a very positive point of view. If you could really address this would be very positive, but nanotechnology is unfamiliar. What about the unintended side effects? How effective would this be? So we decided to engage with publics to build a card game called Demox. This is something I'd actually been involved with since about 2001, with a guy called Perry Walker who invented the idea. We devised this originally for human stem cell research, but it's now being used in a very wide range of issues. And there's a very good website if you want to follow this up. It's a semi-structured group discussion with cards as the medium. You have 6 to 8 people who are dealt hands of cards, make selections, and discuss the issues based on the cards they've got in front of them. The idea is you don't need a facilitator. The experts are the cards, and they can produce their own statements and then vote on different policy options or applications. It comes as a free boxed kit or downloadable, and the idea is it should be played by any group of people, your friends in the pub. Problem: COVID-19 makes such gatherings very difficult. So what we're now trying to work out is, can we turn this into something we could do online? Watch this space. Here is the cards of a typical game spread out on the table. And here is it actually being played at a summer school at Twente, in fact, some years ago. You start off with what are called story cards, where a typical person involved with the technology gives their story with a dilemma at the end. So in this particular case, we have a heart attack patient describing what happened, waking up after their heart attack with all these tubes and so forth, and having to change their whole lifestyle. But then the doctor saying, we want to do a trial with nanoparticles to see if we can detect this happening in my arteries in future. And the question is, should I take part in this? And we also talked the stories of another person who would be the carer of a stroke victim, another one who would be the CEO of a startup company that's got to phase 2 and it's running out of money, and the military get interested. Do I go down the military funding route, or do I stay medical? Trying to do real world ethical questions that actually arise in the technology. And then we have what are called information cards, which give you summarized bits of information about the technology, and then issue cards with all sorts of ethical issues. So how can we be sure that nanoparticles will only go to the intended parts of the body, for example, a question somebody might ask. And as the discussion goes on, the person who is acting as dealer asks people to gather their thoughts together and state an opinion, which we have on what is called the cluster card here in the middle, and which cards they used as a kind of group opinion, trying to get consensus. And then each person as an individual in this particular game gives their view on, here are 4 applications of nanotechnology in medicine. Would these be acceptable to you or not? With the answers, yes, possibly, don't know, doubtful, no, and why, in your own words. And we chose 4 examples: a diagnostics, the imaging of atherosclerosis, drug delivery, and remote monitoring. And we also asked some questions about, would you agree with these statements about risk, about animal testing, clinical trials, and long-term predictions? And just finishing off the talk, here is a quick summary of the results we got back. These games are not intended to be sociologically valid, in the sense that the group is a group of people in society. It's not 50% male, 50% female, etc. But if you play enough games, you do actually get a general picture. And so these are not really quantitative data, but they do give you some indication. And certainly when it came to imaging atherosclerosis, the yes was a pretty strong view. Similarly with drug delivery, a bit less on remote monitoring. Predicting medical conditions was a lot less accepted. When it came to agreeing with statements about animal testing is essential, that got a very strong yes. Taking part in clinical trials is important, yes. Knowing my probability for future serious diseases was by no means certain. And risks outweighing benefits, well, the answer was, I don't know. Because not surprisingly, we hadn't given the information to provide. So you can get an idea of what people think in terms of their views on a subject like, in this particular case, atherosclerosis. But the real point of the game is actually to play it. So that is a quick summary of how we've tried to bridge the gap, that bridge between publics and the technology. And if you want to know more about the game, then please contact me. Okay, that's the end of my talk.
Thank you very much. Thank you very much, Donald. Please stop sharing your screen. Okay, let's go back and let us greet Rafael Levy. He is our next speaker. He obtained a PhD in physics, and interestingly, more than 10 years now, he is one of those few people who are challenging the misconceptions in nanomedicine and nanotechnology in general. Rafael, please go ahead.
Thank you. Thank you so much. So I'm very pleased to speak in this meeting, and I'll try to stick to my allocated time. And so the title of my talk is, Are we building on the shoulders of giants or on a nanobubble? And it's a quite interesting session, really, to be in. And it might seem like these three talks before mine were, going in very different directions. But in fact, I'll try to make some links maybe between all of these talks. So let's get started. So nanobubble. What do I mean by nanobubble? Well, I mean, obviously the word speculative bubble might kind of mean something to you. So, the idea is that there is a kind of an explosion of information or speculation. It could be about a particular technology. It could be about property in a certain area and so on, or the market. Then at some point, it kind of explodes. And I think whether it is a nanobubble or not, there is certainly an enormous number of publications in this field of nanomedicine, nanotechnology. And so we can at least ask the question of how much of this is kind of speculative publishing, if you want, and how much of it is really kind of interesting and valuable research. And then, are we building on the shoulders of giants? So this expression is a very old one. Here you can see a 1200s stained glass that shows someone, the name escapes me now, but on the shoulders of the prophets, enables to, and it's based on a very old saying, so of this period, unable to see further. In this case, it's a kind of religious reference, but in the scientific context, it's basically the idea that we're not doing science just based on our own results, experiments in the lab. And here I kind of comes back to Lou's definition of science. We build on what everyone else has done before. And that's the way we can do that in the current period is by looking at the publications of our colleagues, mostly in online journals, and of course, older records as well. So how much of that is basically solid? And are we building on the shoulders of giants, or that there are a few questions that we need to look at, or in a nanobubble? So the first thing is that we can't build on something we don't know, or even something we don't want to know. And here I'm going to give you, I've mentioned culturally induced ignorance here, and I'm going to give you some example of what I mean. Do nanoparticles cross the blood-brain barrier? When in the session before the break, before our session, the last talk was by Alejandro Sosnik, who was basically trying to solve this problem of getting things across the blood-brain barrier. And, it remains a very, very difficult issue today. Yet, the big fuss about nanoparticle crossing the blood-brain barrier started in the late 90s and early 2000s, and was one of the drivers for the kind of explosion of nanomedicine. And it was reported that peptides, which are kind of bound to colloidal polymer particles, could cross the blood-brain barrier. And that was quite a strange discovery, because it was already known at that time for many years that if you look at how much things go through the blood-brain barrier, as a function of their size, well, I mean, when you are in the kind of nanometer range, you're already way too big to just diffuse through the blood-brain barrier. So it was quite strange, and it was actually contested a few years later, but still, this paper is very heavily cited. This one is not. And here we are now, 25 years later, and this is still a very difficult problem. It's not something that nanoparticles have solved, certainly not. And why do I say that it might be something that we don't want to know? You know, we might prefer thinking and talking about particles crossing the blood-brain barrier, at least for a number of years, until we have to face the fact that really, actually, it's really hard. Because actually, when we write grants and we try to publish papers in Nature, Biomedical Engineering or elsewhere, it might be easier to say, look, we've got these wonderful nanoparticles that are going to cross the blood-brain barrier and we're going to solve problems, rather than something else, like negative results, for example. So we might not want to know, for example, when we apply for grants and when we write introductions of our papers. Nanoparticles readily travel throughout the body and penetrate cell membranes. I mean, you know, this is relevant also to Donald Stoke, because certainly one of the reasons for the concerns about toxicity is also an exaggeration of that idea that, you know, nanoparticles are somewhat magical and they can go everywhere in the body. And that sentence, you know, nanoparticles travel readily through the body and also they can just penetrate the cell membrane. That sentence is from a science paper, a science review from 2006 that has been cited several thousand times in the literature, but also that has been cited in many, many reports, you know, from the European Union and elsewhere. So these kind of exaggeration are quite well spread and have an enormous impact on how the field is developing. And this is quite surprising if you think that in the 1950s, it was already known that nanoparticles enter cell by endocytosis and end up in endosomes. So, you know, these pictures show gold particles in endosome, dates from 1957, and gold particles were even kind of one of the contrast agents. They were not called gold nanoparticles, they were called gold colloids, of course, at that time. So we might not want to know, we might prefer the idea that nanoparticles are somewhat magical and can go everywhere into the body, because that makes more buzz and therefore more grants, more papers. And of course, we can't build on something we don't want to know, and we also can't build on something which is wrong, which brings us to the question already also alluded by Lou, which is how much of the scientific record is accurate and reproducible. And you mentioned a report from the Netherlands from 2 years ago, I think, saying that, you know, it should be a normal thing that replications take place. But, you know, in nano, I'm not aware of any straightforward, like, direct replication paper that's called like a replication. So it should be a normal part of the work, but actually it doesn't happen. So we don't know the answer to that question of how much a scientific record is accurate and reproducible. Then we have this idea that science is self-correcting, right? We agree on that, everyone? I would say obviously not, right? So obviously not. First, obviously not. Absolutely not in the sense that science is not doing anything. Like, I'm doing things, you're doing things, scientists are doing things. Science is not doing anything. So in that sense, science is not self-correcting. It's only going to be self-correcting if people try to correct things. And then if the system is such that self-correction is actually plausible and happening. So let me tell you very briefly about some experience I have with attempts at self-correction. So I have more actually, but I'll go extremely quickly on this because I could speak for an hour for each or more. So the first one is from 2004, where there was this paper, it's scanning tunneling microscopy reporting stripy nanoparticles. And basically, I thought that it was an artifact of scanning microscopy. And a couple of years later, because there were more papers coming through, I tried to correct this. I'll just basically say that this story took several years to publish our paper, which was called Stripy Nanoparticles Revisited, eventually turned a little bit into a controversy, which got a little bit of coverage in various kind of science-orientated publication, as you can see. And I'll skip directly to the lessons of that story, which is basically that This was hard work. It was hard work, was slow, depressing at time. And the most important thing is that scientific journals are very reluctant to correct the scientific record. They want the buzz, they want the impact factor, they want these kind of things. They're not particularly interested in correcting the scientific record. Even institutions, their job is to protect their own reputation, not the scientific record. So they're not going to be particularly helpful. Access to original data is key. That's why, I was pleased to see that Lou's insistence on sharing of data, for example. And then I do think that blogs and social media, which have been mentioned in kind of a negative way earlier, blogs and social media help in that process. Where traditional scientific journals fail, blogs and other social media provide a new space where science can be discussed in a much more rapid way, where you don't have to wait 3 years before a paper is published, if it is ever published. I'm going to give a few more ideas of things we can do. So the second story is spherical nucleic acid. So that's from Chad Merkin at Northwestern. I'm sure everybody knows Chad Merkin. And basically, again, from one of his papers, you can get particles enter freely into cells. That starts with a paper in Science in 2006, and then many other papers on these particles covered with oligonucleotides, which are supposed to detect mRNA inside cells. And the problem is actually exactly the same as the one I was mentioning before. You can't detect mRNA inside cells if you are inside endosomes, inside these vesicles, these bags inside the cell. You've got to get outside of these endosomes. If most of your particles are in endosomes, which they normally are, then it cannot work. So the reason I was interested in that story in particular also was that in that case, replication was relatively easy because the technology was commercialized in 2013. So you could buy the particles and try to use these. And I was based at the time in a biology lab as well. So I had people who were interested in detecting mRNA inside cells if it could work. So we basically tried, concluded that it didn't work, published our results on my blog and on another kind of alternative science platform because there was no, it was going to be extremely difficult to use the traditional route. And then I got this email from Luke Armstrong, who was someone working in the company selling this product for several, and his job was to actually make it work and develop applications. And he basically left the company because the technology did not work. And they continued to sell this stuff for several years, basically for 5-6 years, they continued to sell this stuff. And it must be that many authors, many scientists tried to use it and got negative results and didn't publish it, which brings me to the next slide. I very strongly recommend the Upturn Microscope, which is like a cartoon, science cartoon site and author. And so, he's quite unhappy that blue guy is what's wrong. I've been testing this stupid protocol for over a year now. It doesn't work. I've wasted my entire grant. I have nothing to show for it. Let me see that.
Sorry for interrupting you, Raphael. You have 2 more minutes.
Thank you. I'll try to finish in that time. I remember this. I tried it over a year ago and gave me negative data. Since I couldn't publish, I just filed it away. Never told anyone. Doesn't end well. Negative data is still data. Publish it for everyone's sake, and you can do it. So, for example, I had this paper which is negative data. It was rejected by not by Rosie herself, but by her colleagues at Nature Biomedical Engineering. But ChemArchive is there, so we can just publish it as a preprint. And these numbers are actually of views and downloads are from quite a while ago. I haven't updated it, so people can find it, people can cite it if they want, and they certainly can read it and know the argument. So we need to find ways of improving things. One way is post-publication peer review, and we can use Twitter. We can use PubPeer, which if you don't know about PubPeer, you should all have a look. You can use preprint. So we need to basically open up the space for discussion. Of course, you can also use Precision Nanomedicine, this excellent journal, isn't it, Lou? So I've just published a paper there, which is kind of a perspective. If you're more interested in this issue of the nanoparticles crossing membranes and why there are misconceptions on this topic, which are persisting, I invite you to have a look at this paper. He's waiting for my correction of Galli's proofs, but there is already a version which is online. And with that, I would be very pleased to take any questions, and I thank you for your attention. Maybe just one more word, if you allow me. If you're interested in these issues and you would like to contact me, please do follow me on Twitter or send me an email. I have quite a big announcement coming up next week. I can't tell you more, but if you're interested in these issues of replication and so on, and correction mechanisms of science, please get in touch. Thank you.
Thank you, Raphael. And there are a few questions. Actually, there are two questions, and I asked both of them. And let me change my view. So standard speaker. Okay. So there are no external questions at the moment, but I do have one question for you, and then one question for Rosie as well. And of course, every one of you. Let's ask those questions at the very end. So our next speaker is going to be Beat, who is organizing these great meetings, things like 12 years. This is the 12th one, and I was lucky to participate starting from the second one. Sorry, I missed the first one. I didn't know about it. So, Beat, please go ahead. How do you manage all these things? I'm very curious.
Well, can you hear me? Yes. Okay.
Yes.
I think I wrote the title and said, I take the Klingon perspective. And maybe I go a bit back and say that 1994, I created a company. And when they asked me, what are you doing? I said, I do concept engineering. And they said, what is that? And what I said is, there are many people who walk around with visions, but they have no notion of what their goal is. And they have no notion of what actually, how they would do it and what the mission would be. And then I said, I'm specialist on telling you what is your mission, what is your strategy, and maybe I give you a set of goals, and then you can say what is my primary goal. So the next step was that I have found a client who asked me to bring together the University of Freiburg, Basel, and Strasbourg, because they wanted something like a bio valley like a small Silicon Valley for biotechnology. And I worked for 6 years with the team, and it was very difficult to change minds of people, and it was only possible due to the fact that these people got into a debate together and understood finally from one from the other what the goal is. And later I worked for a while for a Japanese company and as a business developer and for research management responsible. And I can tell you, when you work for a company with a different set, it's enormously important that you understand that probably you can change nothing. And when I came back from this, from Düsseldorf to Basel, I went to a conference and I heard about how fantastic it is in Basel with nanophysics and with medicine and with pharma. And someone asked me, what are you going to do now? And I said, oh, I'm going to make the hugest network in the world. You don't have to take small goals. You have, if you have a big goal, you maybe you maybe can do something small. And I said, well, it's clear. I'm going to make the biggest and largest network in nanomedicine. And he connected me with Patrick, and I saw Patrick Hunziker, who is a clinician. And very soon we said there must be a clinical part, and there must be a part to set up the largest network in the world. So we started out with 100 people, with 150 people, with 200 people, 300 people, 400 people, 530 people. And what actually was the goal? The goal was that if you have excellent science, if you have young and old people together, that this mix and a wonderful ambient and the impression that you are a family, then you start to trust and to talk to each other. And this is what happened at Clinam. Everyone who comes there doesn't care what the record is or what is right or false in the journals. He comes to speak to other people, to find a way how to debate and to understand each other, and similar goals. We could do something together. We have made 5 European Union projects within Clinam. There are 5 startups that still exist. There are 28 corporations that were sets of cooperation that were established. And this is not due to Clinam. Clinam is the platform. And we know how to host the people, and we know how to make them happy. Because being happy is the ground setting for being able to do a good thing. I was always told that research is scientific research innovation. Innovator, I'm a research innovator. It's stupid. No, it's not. Innovation happens, and it happens by people who are engaged, who love their job, and who love others that they meet to cooperate. And this is what Clinam is. Clinam strives for conferences of excellence. And now let me tell you that I was very successful with these conferences in real life. And this year, about 2 months before this summit started virtually, I was getting up every morning at 2 o'clock thinking, I know I'm going to make a virtual conference and no one will be coming. Because virtual is so difficult and is so distant. So we tried to make a website where you see films, where you have a virtual evening event, where you have a dwarf award, where you have this and that. And we tried to bring a bit stimulation into it, that the people say, hey, this is also life. And not only science, but this is Clinam. And to bring together these people was great. And today I was very astonished because I learned something that 460 people were online today and have listened to you and to me and have been here. And this is incredible because I think that shows that there is a need beyond publishing, beyond peer reviewing and re-reviewing, that people meet, people come together and learn from each other. This is the best communication that we need in order to make progress. And then innovation happens and acceleration is given. Thanks. That's all.
Thank you very much, Bert. It's great to hear the story. You know, it is trivial to you, but only because you were there in the middle of every time, and it's good for everyone, especially the new members, to listen and hear the story of the meetings. So we have like 3 questions. Can we switch back? Can you guys hear me?
Yes.
Can you guys see me?
Yes.
Excellent. Thank you very much. So the first question is about urban myths. I think it's kind of open to every one of us that say the question sounds that greenhouse gas emissions from agriculture are widely believed to be as high as an impact from transport, from cars and so on. The authors acknowledge they made a mistake in their methods, but this has not been well publicized, and the whole thing became an urban myth. So what can we do?
Well, do you want me to comment on this?
Sure.
I mean, I wouldn't comment on the greenhouse emissions, but I think when we talk about urban myth, there is a danger that people listening to us might think that we're talking outside of the science. So we're not talking about peer-reviewed publications, for example. So I'm particularly focused on peer-reviewed publications because there is an idea that science is somewhat different, because there is a scientific methods, we have peer review, we have standards that we are supposed to operate with, and therefore urban myths basically should be more or less removed by these methods and standards and practices. Yet what we see is that not only they're not removed, they're amplified by many of our practices. Like there are things which are pretty wrong in nanomedicine and which are repeated in hundreds and hundreds of introduction of papers. And I'm sure even in some of the talks happening at the Excellent Cleanup meeting. And so the question really for me is, why is that happening? And also, what can we do about it? And I think that some of these things depend on the kind of field of science. But at least in my field of science, I don't have the feeling that robust contradiction is something that is welcome or normal. Quite the opposite. When I ask questions, if they are a little bit critical, the reaction can be relatively violent. And then, of course, nobody else is going to ask a question if that is a reaction. So the dynamics happening here is one I think we need to work on.
That's all right. So we have like 3 minutes left from our time before the other session starts.
Well, I would answer to Raphael. It's all about communication. If you propose that there is a mistake, it's much more accepted than when you oppose in a way that the other has no chance to chase away.
Can I respond to this briefly? I quite disagree with that. Of course, we have to stay professional at all time. We have to make sure when we disagree that we make our argument very strictly about the facts and the evidence. But even if you are the most polite, if you, for example, think that another scientist's work is wrong, let's just make things very simple and clear. Let's say you are proposing very politely that a dozen of papers by one of your colleagues is wrong. Even if you do it very politely and with a big smile, that other colleague is not going to feel particularly pleased about it. And there is nothing that you can do, in terms of how you communicate to make it pleasing. Of course, that person is not going to be pleased about it because that's something they've invested a lot of energy. Maybe they've partly built their career on it and so on. And they might have, Lou talks about conflicts of interest. Obviously, there are big conflicts of interest. Their career might depend partly on some of these things. So I don't think it is. I don't think it is as simple as just being polite. I think that for science to progress effectively, we need to have spaces where these discussions can take place, where the evidence can be debated, even when there are conflicts, and these conflicts need to be managed. And at the moment, I don't feel that these spaces are very welcoming. And of course, there are power dynamics here. I mean, if you are a PhD student and you think that your boss or even someone else, a boss in another team, has made a big mistake, you might kind of hesitate before you say so, because your power in that relationship is not quite the same as the power of the person you're challenging.
And that I agree with you.
Yep.
Of course. I just think what happened to me in the last 2 years is that I have two scientists and they have a war together, and they have always consulted me all the time. One has told me his story, the other the counter story. And in the end, it was very good. We found a solution how to sit around the table and to discuss. And it was interesting to see that two people who are going totally against each other have found a solution together. I don't think this is a good example, maybe, but yeah, generally I agree that it's in our society it's very difficult to change things and to say things, not to be cut off.
Well, thank you. And I suggest that let's go over to Debate Lounge 1, which is a different Zoom call, and continue if you wish. There are two more questions. And of course, this discussion is not closed at all, but we have to give space to the next session. Okay, so see you there in like a minute.
Okay.
It will be Chezi Barenholz in his plenary session.
Yes. Thank you.
Thank you very much for everything.